on a personal note, I'd like to say really enjoyed working with Ben, and I'm sad we didn't get to do more of that working in person, but I'm looking forward to seeing all the great things he'll accomplish in the future. So without further ado, I'll just let Ben present his project on cardinals, grosbeaks, the evolution of them, the evolution of color and habitat. So take it away, Ben.
Awesome. Well, thank you, Kevin, for that introduction. I guess another way you could describe all the work I've done is maybe compulsive. But at any rate, today I'm really excited to talk to you all about all the work I've done over the last 3 years. And specifically today, I want to tell you a story about why organisms look the way they do and how human biases have shaped our understanding of those appearances. This is kind of be formulated in the idea of the conventional paradigm of sexual dichromatism, which dictates that sexual selection pushes males to be vibrant and colorful to attract mates, while natural selection pushes females to be cryptic to blend into their surroundings. However, this binary description of color can be really misleading, with formulating that female coloration is not as important as male coloration, and additionally ignoring the broad mosaic of variables which influence coloration patterns. Coloration arises from a complex concert of selection pressures, a litany of causal links between different life history traits, mating systems, habitat structure, and climatic variables influence how color evolves and perpetuates. So for example, migration has a strong influence on plumage coloration, as species that migrate tend to be more sexually dichromatic. There are two competing theories on why this is. The first one is that males are more colorful to deal with, to show off their success after a long flight. Whereas more contemporary research, not better, but more contemporary research suggests that females are drabber to deal with the associated perils of a long flight. And that can be visualized or surmised in the migration cost hypothesis. Additionally, climate has a really big impact on how color evolves, and Glauger's rule describes a very broad ecogeographic theory of how organisms tend to be more heavily pigmented and darker in humid environments, and tend to have less melanin pigmentation in lighter environments. This is largely driven by the trade-off of melanin deposition and how it interacts with precipitation and temperature. And then there's a couple additional variables, such as how microclimate influences darker feathers to be a useful tool to defend against parasites, to regulate temperature, to camouflage. But for the purposes of this talk, we'll really focus on those broad scale patterns. And then finally, one of the most important abiotic features driving plumage coloration is light environment. Several studies using categorical metrics, such as open and close, have found that the amount of light in a habitat impacts signaling modality and how species can perceive one another. This drives a species' plumage to either resemble the ambient light conditions of its habitat or evolve conspicuous plumage to stand out. And what that really just translates to is that birds in darker habitats are darker and birds in lighter habitats are lighter. Additionally, some other studies have found that there's a higher diversity of color palettes found in closed habitats. However, when we're using these tools, we've often used categorical metrics such as open and close, and those are really informative to paint a large picture of how habitat shapes coloration patterns. But habitats aren't simple binary descriptions. They're these very complex mosaics, and it is very difficult to draw a line when a habitat is closed and when it is open. A few studies have attempted to have a more robust metric to define habitats. Friedman and Rees found that across all Australian birds, climate drove birds to be darker in the tropics, but they actually found that open regions had greater color span than closed habitats. Additionally, Medina et al. Found that habitat structure was really correlated with female coloration, but not male coloration in the fairy wrens. And these two studies point out that these ecogeographic patterns are playing a large role in how plumage evolves, but the relative importance of them in initiating and perpetuating patterns of plumage evolution remain understudied. And furthermore, to date, no studies have attempted to see how these ecogeographic variables impact plumage in a sexually dichromatic family. The cardinals are a really excellent group to tackle this question because they have varying degrees of plumage morphology as well as differing degrees of sexual dichromatism. Approximately 1/5 of the cardinals are migratory, And most cardinals are sexually monogamous, which means that conventional metrics which drive elevated levels of sexual dichromatism, like polyamory or lek breeding, are not applicable to the cardinals. In addition to all of this, the cardinals also occupy a very wide range of niches across most of the habitats. For example, the rose-breasted grosbeak lives in temperate boreal forests. Perluxia lives in desert scrub and mesquites. The rose-breasted chat lives in subtropical dry broadleaf forests. And the red and black grosbeak lives in tropical lowland humid forests. So taking all of this together, I was really curious, how has natural history and traditional habitat metrics influence patterns of sexual dichromatism? And how does habitat heterogeneity influence coloration patterns? So to tackle this question, I developed these 3 chapters to really get at the heart of it. And the first one being a phylogeny of Cardinalidae based off high-throughput sequencing of ultraconserved elements. So there is currently a phylogeny published by Barker et al. In 2015. This was for 44 taxa and was done using 4 mitochondrial genes and 2 nuclear genes. However, even the most conservative taxonomies suggest that there are 49 recognized species, and multiple nodes within this phylogeny are poorly resolved. So I set out to create a new phylogeny using 68 taxa. UCEs are really popular markers that have proven to be effective at resolving taxonomic uncertainties at multiple timescales. Each UCE is characterized by a large conserved core region, which doesn't vary dramatically, but on the outsides, we have these 2 approximal ends, and these approximal ends accumulate more variation over time. These distributions can be referred to as a smile curve, and we can use that variation that they accumulate on those approximal ends to compare interspecific variation across deep time. So I had 4 different sampling metrics for my taxa. The first 2 I extracted on my own. I extracted DNA from 45 liver and tissue samples using a QIAGEN DNA EasyKit. And when tissue samples were unavailable, I extracted DNA using a phenylchloroform, which I then quantified using a qubit. I then outsourced this data to Arbor Bioscience, who sequenced and performed the library prep. 14 UCEs for the genus Paranga were previously published by Mapley et al. In 2016. So I downloaded the raw reads from NCBI and incorporated them into my phylogenetic pipeline. And then finally, for outgroups, I used Theropidae, the sister family to the cardinals, and those were published as part of Rosalind Price Williams' thesis in 2019. So I took all 4 of these sampling schemes and brought them in to Phylogeny. And Phylogeny is a multi-program platform that is really meant to prepare phylogenetic data for downstream inference. And there are a lot of programs and a lot of steps that are part of this pipeline, but I'm really just going to kind of go over the basics of what it does, which is trimming low quality bases, counting and cleaning reads, assembling contigs, matching those contigs to a previously published probe set, and aligning your sequence and trimming that sequence. And after all of that, I recovered about 5,000 loci with an average of about 1,000 loci across all taxa. If you're familiar with UCEs, you understand this is actually really low. And that's because the topads failed dramatically. I covered on average 1,700 contigs per sample with a very low average length, but across all these, they had differing degrees of coverage. To give you an example of how bad this is, the tissues on average covered about 4,200 contigs with an average length of about 1,300. And this is a huge issue because topological inference can really be impacted by gene alignment lengths, where shorter gene sequences can produce higher gene tree estimation errors, which will generally just worsen your phylogenetic estimates. So I had these 5 different taxa with differing degrees of coverage, yield, size of their contigs, and just effect that they would have. And I really struggled to figure out how I wanted to move forward with these samples because I didn't want to include samples that might bias my downstream results, but at the same time, a lot of these had not been included in a phylogeny before. So ultimately, what I decided to do is to remove the worst individuals. So in my final data set, I had 66 taxa, And in my 75% complete matrix, I had 4,200 loci. However, to ensure that these individuals were not having an effect, I ran all of my analyses with those low coverage taxa and without those low coverage taxa. For my phylogenetic inference, I had two major methods that I used to analyze my data. First, a concatenated metric using maximum likelihood. In this, I used IQ tree. I used, for my model of evolution, I used GTR plus I plus G using 2000 ultra bootstraps. I partitioned using model finder, and within IQ tree, I generated gene trees. And using those gene trees, I calculated gene concordance and site concordance factor. And these are just alternative ways to really understand the underlying variance in your concatenated metrics. And they're just an additional summary statistic to bootstraps. And then finally, I analyzed my data set using a multi-species-coalescent based method. And for this, I used SVD quartets. I don't have the time to go into all the results of all of these trees, so I'm really just going to showcase my final tree, which we can sit here and appreciate for a second. It's very beautiful. I'm very proud of it. So I found convergent results across all concatenated and coalescent based approaches with nearly 100% posterior probability and bootstrap support across all nodes. All of these splits were further validated with gene and site concordance factors. And I found that the low coverage taxa didn't seem to really influence the topology, with the exception being in the coalescent based approach, those low coverage taxa appeared arbitrarily across the tree, but when they were removed, I recovered the exact same topology as the other coalescent and concatenated based approaches. The results of this are generally concordant with the Barker et al. Phylogeny, with the exception of two key instances. First, Amuraspiza is not actually sister to Cyanolaxia, but instead sister to Passerina. And Habia rubica is actually not closely related to the other Habia species, but is actually very distantly related to them. So in summation, UCEs resolve conflicting nodes and establish new phylogenetic relationships within Cardinalidae. Now I can dive into my second chapter, which is assessing the impact of habitat and migration on the evolution of plumage coloration. I was really privileged to be able to travel to museums across the country where I really got to see the very wide berth of all of these different data sets and specimens that have been collected over the past 200 years. And I quantified color using a spectrophotometer, which is measuring the exact wavelength of light that is reflected back across any particular patch. And I had to do this because birds have a different visual system than we do. So in order to understand how plumage is evolving from their perspective, we need to model it from their perspective. So to do this, I use the spectral sensitivities of the blue tit to calculate all 4 quantum cone catches. Those are the ultraviolet, long, short, and medium wavelengths. And then I took that data and plotted those reflectance spectrum within tetrahedral color space. So here, each of those reflectance spectra are plotted at the achromatic center of the tetrahedron and then moving out and ending at any particular region. And within tetrahedral color space, we can not only model color using the avian visual system, but also calculate statistics that are describing coloration. And these include color span, which is within plumage contrast, volume, which is diversity across all patches, hue disparity, which is disparity across all patches, chroma, which is saturation or how different the color is from black and white, and brilliance, which is how light the bird is. For each specimen, I measured 17 standard plumage patches, and for each patch, I measured it 3 times and averaged that to have a single metric. For each species, I measured 5 males and 5 females, which summarized into a lot of time sitting in a museum looking at a lot of dead birds. But ultimately, what this resulted in is a data set of a lot of different traits describing how plumage color looks. And I use a phylogenetic PCA to phylogenetically control that data to create single axes describing male and female plumage. For this, I actually used the Barker et al. Phylogeny because my UCE phylogeny had not been completed. So for the results of this, I found that PC1 is loading highly for all traits except for hue disparity. And it's really describing a gradient from drabber, darker plumage to highly colorful and complex plumage. And PC2 is loading entirely for hue disparity. So this is really describing a gradient from highly contrasting patches with lots of blacks and blues to more monochromatic species. And then for females, I found a very similar pattern for PC1, where PC1 is describing a gradient from conventionally drab plumage to highly colorful and complex plumage. And PC2 is loading mostly for chroma. And here, positive values represent more colorful species and negative values represent species that are darker with more blacks and whites. Additionally, brilliance did not load highly for the female PCA, so I analyzed it separately for all my downstream results. To quantify ecology and habitat, I pulled a lot of different data sets from different sources. And the reason I did this is because I was really curious how user-defined metrics of habitat influence how we interpret our results. So I first collected data from Barker et al., and here I collected data on foraging strata, and I broke habitat down into a category I will refer to as non-forest. So do they live in a forest or don't they? My second data set is a more fine-tuned description of habitat, which came from Guiar et al. 2021. And here habitat is broken down into semi-open, scrub, and forest. Then finally, I collected data from BirdLife, which includes data on migration and species dependency on a forest. So my hypotheses for this are based on previous literature. The first being that evolutionary changes in both sexes will drive elevated levels of color complexity. The second is that light environment will cause species to be darker in darker habitats and lighter in lighter habitats. And finally, I predict that there will be greater color contrast in closed habitats. And to analyze the correlated evolution of male and female plumage, as well as plumage and these different habitat metrics, I implemented a phylogenetic generalized least squares framework under a lambda model. And right off the bat, I found that male and female plumage were highly correlated with one another, implying that both sexes contribute and reciprocally influence elevated levels of plumage complexity. Even though PC2 loaded for different traits for males and females, I still regressed it and found a slight negative correlation, although there is no phylogenetic signal, which means that it's just a regular regression. And as I said, I analyzed brilliance separately, and brilliance between males and females was highly correlated, and this is likely due to shared selective pressures that these species have across their different habitats. And now I'm going to dive into the results of how habitat influences coloration. I'm not going to show the summary statistics tables. If you are really interested in those, we can talk about them at the end of this talk. For females, I found that they were predominantly influenced by ecological traits. So the foraging strata occupied and migration. And what this breaks down to is that a model for female PC1, a model incorporating migration had the most likely AIC score, and specifically sedentary behavior. And likewise, for female PC2, I found that migration and sedentary behavior were correlated and had the most likely AIC score. And finally, for brilliance, I also found that understory and the strata occupied was produced the most likely model. So what all of this breaks down to is that females are drabber if they are migratory, and if they occupy the understory, which is a low light environment. Whereas females have evolved to be more colorful if they are sedentary. For males, however, I found that habitat was really influenced with coloration patterns. So for male PC1, again, this axis of plumage complexity, I found that a model incorporating habitat had the most likely AIC score, and specifically semi-open habitats, where males have evolved more complex plumage patch patterns if they occupy the semi-open fragmented forests. For male PC2, I found a really similar pattern, but instead, a model incorporating forest dependency had the most likely AIC score. And here, the effect was medium dependent forests. So taking these two results into account, it seems that males are evolving more complex plumage with more contrasting patches if they live in these kind of semi-open fragmented forests. And this is likely so that they can stand out in these habitats. This is also some brilliance is also showcasing this pattern, as I found that the strata occupied was really correlated to the understory. So what that really just means is, similar to females, darker birds live in darker habitats. An interesting note is that migration was associated with most of these models, but it was never significant. That implies that while migration is associated with these really complex patterns, it is not correlated with it. And this presents really interesting evidence that female coloration is really influenced by migratory behavior and male coloration is not. And that's a very, very strong evidence for the migration cost hypothesis. And so this now paints a really nice picture that females are really interested by ecology and males are really interested by habitat structure. But now let's take a deeper dive into the actual statistics of these. So again, for habitat and specifically semi-open habitats, I found that male PC1 was highly correlated with these semi-open habitats. But I found the male PC2 was not associated at all with semi-open habitats. And likewise, for forest dependency, I found the PC1 was not correlated with medium-dependent forests, but was highly correlated, or excuse me, was not correlated with semi-open habitats. I was right the first time, but PC2 was highly correlated with medium-dependent forests. And this is really concerning because these two habitat metrics should inherently be describing similar patterns, but they're not. All three of these data sets come from peer-reviewed journals and data sets. So forest dependency comes from BirdLife, which uses the IUCN classification, habitat classification scheme. Habitat comes from Villar et al., which interpolated data from Birds of the World, the premier ornithology database. And non-forest comes from Barker et al. And that really begs the question, how do human biases influence our habitat designations? And it really seems that how we choose to define these categorical metrics to try and squeeze habitat into a single variable really impacts our downstream results and how we interpret those results. So in conclusion, I found that there are alternative selection pressures on both males and females. For females, I found that they are heavily influenced by strata and migration. But for males, I found that migration doesn't really seem to influence plumage complexity, but instead, males have evolved highly contrasting plumage patch patterns in these semi-open habitats. And that's likely, again, to help them stand out in these forests. But all of these results have a large asterisk next to them because it seems that character selection has a very large impact on our results. So it seems that when we are using these categorical metrics, we are quite literally missing the forest for the trees. And it really begs the question, how do we actually quantify habitat? And that's what I'm hoping to get to in my third chapter, which is a large-scale spatial analysis of the joint impact of habitat and climate on the evolution of plumage coloration. I have a very large pipeline for this chapter to untangle how all of these ecogeographic effects influence the evolution of coloration patterns. So I'm going to go through this as quickly as I can. First, I needed to incorporate data from my first and second chapters. I now have my UCE phylogeny, which I time calibrated using the Kronos function in ape and the divergent states from Barker et al. This allowed me to add 3 new species. I then incorporated average mass, which I log transformed. And then finally, I had to reanalyze my color data in using a non-phylogenetically controlled PCA because I really needed to make sure that male and female plumage were on the same axis of comparison. This is what the non-phylogenetic PCA looks like. The results are pretty comparable to the non-phylogenetic PCA. PC1 is still describing a gradient of overall plumage complexity, and PC2 is really describing a gradient from chroma to average hue. And I've colored this now by PC2. And so you can see the difference between species that are darker at the bottom and species that are more colorful at the top. Another way we can break this down is by sex. The red dots represent female species that are located in the upper left-hand corner, and the blue dots represent male species that are kind of distributed across the right side of the graph. Additionally, I regressed the female phylogenetically controlled PCA and the female non-phylogenetically controlled PCA. and I found that they were very convergent with each other, which means that this non-phylogenetically controlled PCA is really just correcting for male plumage, and it's doing that by incorporating female plumage. So now that I have my corrected color values, I can start to generate species distribution models. And to do this, I downloaded and cleaned occurrence records from GBIF. I did this to remove common spatial and temporal errors, and this includes tasks like removing superfluous, duplicated, or inaccurate records, as well as removing records from areas of high human density. I then spatially thinned these records using kernel density estimation and a threshold of 65%. And I did this to mitigate the effect of North American species. There's a very interesting problem in community science data where North American species are grossly overrepresented and South American species are grossly underrepresented. And so I wanted to ensure that any downstream analysis that I was doing wasn't being influenced by those the heavy presence of North American species, but I didn't want to influence the very low accounts of those South American species. So I used kernel density estimation to generate unknown probability densities, and kernel density estimation additionally allows those points to retain their spatial positioning. So using those probability estimates, I then thinned my records to only retain 65% of the data. And then I used that to generate species distribution models. Ensemble species distribution models are weighted average species distribution models incorporating multiple algorithms. These syndicate these multiple algorithms, and using an average and an uncertainty threshold, which is just between model variance, it generates mean habitat suitability. So the two algorithms that I used were the Mars algorithm, which is an extension of linear regression that automatically models non-linearities and interactions between covariates, and the CTA algorithm, which is a machine learning classification algorithm that uses recursive partitioning to just break the data down into smaller, more homogeneous subsets. And so now that I generated these species distribution models for all of the cardinals, I then created binary maps of occurrence, which I uploaded to the Google Earth Engine. The Google Earth Engine, briefly, is this incredible repository of geospatial and remote sensing databases that allows a very easy platform to analyze these databases, all using Google servers. And so I use the Google Earth Engine to query all of my habitat data. And briefly, the Google Earth Engine is an incredibly powerful tool, but it does set computational limits on its users, which is why I needed to generate those binary occurrence maps to analyze within the Google Earth Engine. So to quantify habitat, the major way that I quantified habitat was through empirical-based vegetation indices. And these are really powerful tools to analyze how community composition changes across both space and time. And they're derived by comparing the spectral reflectance signatures of leaves. And really, that's done by comparing two different bands, the near-infrared band and the visible red band. So briefly, what this means is habitats that are greener reflect higher concentrations of chlorophyll. So that is going to reflect a higher proportion of near-infrared light, but browner habitats are going to reflect a higher proportion of visible red light. So we can take these two spectral bands and correct for the difference between them to create NDVI, which is the normalized difference vegetation index. Now, this is a really powerful tool to analyze how community composition changes across both space and time. What you're seeing here are two different GIFs describing how NDVI changes across a 3-year period from January 2011 to December 2014. On the left-hand side of the curve, you can see a desert-dwelling species, and you can see how the vegetation composition ebbs and flows over time. However, on the left-hand side of the slide, you can see a tropical dwelling species where vegetation composition remains fairly stable over the years. So for my vegetation metrics, I query data on NDVI, its sister band EVI, which is very similar to NDVI, but it just has enhanced correction over the rainforest. And then I queried the two spectral bands that make up those indices, near-infrared and visible red. And so taking these 4 different metrics, I queried image stacks to generate, based on 16-day intervals from 2000 to 2019, to generate this huge amount of data describing how vegetation composition changes. And I took this large image stack and reduced it down using a reducer, which takes our sandwich of data and squishes it down into a single plane. And from that single plane, I calculated summary statistics, such as mean, standard deviation, min, and max. And I was very excited about this tool, but although vegetation indices are really powerful ways to study how vegetation community structure changes across both space and time, it can really struggle to differentiate between highly green forests and highly green grasslands. And this is because both of those habitats exhibit higher chlorophyll concentrations during certain times of the year. So to get around this, I included 2 additional data sets. The first being a global tree forest cover data set, and the second being a data set comprised of land cover types. I'm going to briefly talk about how I calculated both of those. For percent tree cover, every pixel in this data set represents a 30 by 30 meter grid, where each one of those grids is a percentage of trees above 5 meters. So I queried and summed the total number across each species range and then divided by their surface area to generate tree cover per hectare squared. The land cover data set is calculated very similarly. Here, each pixel represents one of 17 different land cover types. I summed the total counts of each of those categories, and then I standardized by each species range and divided by the total. And what this whole process means is that I now had a percentage per land cover type per species. And then I reduced all of that into a PCA. The figure that you're looking at right now is the summation of those 2 different data metrics. It's colored by percent tree cover, where greener values represent species with more habitat or more tree cover, and redder values represent species with less tree cover. Land PC1 described approximately 25% of the variation, but really was capturing a lot of noise across North America, which was not as informative as I was really hoping it was. However, Land PC2 was very informative because it is describing a gradient from explicitly evergreen broadleaf forests to all other habitat types. So now I have this really cool metric that is describing habitat specialists in the rainforest to habitat generalists. And now to quantify climate, I generated hypervolumes for all of the cardinals and then calculated the centroid of that hypervolume. And then I aligned that data to WorldClimate's bioclimatic database, which is 17 different variables describing climate in that particular region. I then took that data, Z-transformed it, and ran a PCA on it. This is a very similar scatter plot to the one you just saw, except now it is colored by latitude, where greener values represent more northern species and redder values represent more southern species. PC1 is describing 60% of the variation and is describing a gradient from seasonal climates to highly stable and rainy climates. And climate PC2 is describing a gradient of precipitation from positive values representing drier climates to negative values representing wetter climates. So then I could take all this data and concatenate it into a single matrix to assess the correlated evolution of plumage and ecogeography. Before I really get into the analyses, I realized that a lot of these habitat metrics were probably autocorrelated with one another. So to deal with that, I removed any traits that had a regression coefficient of plus or minus 0.75. This resulted in 14 remaining traits. And then I took these 14 remaining traits and implemented them in a similar PGLS framework to understand how each independently and jointly affects plumage evolution. And so I assessed all single trait comparisons, all double trait comparisons, all multi-trait comparisons. And then for those multi-trait comparisons, I ran PGLS ANOVAs to understand the relative importance of each of those variables. So now that you understand the theoretical framework of these analyses, I can finally tell you what my hypotheses are, which is that A, male cardinals will evolve more complex plumage and more heterogeneous habitats. And that's based on the results of my second chapter. And 2, I predict that females will evolve drabber plumage to match habitats with higher brown reflectance. So browner females will evolve to match browner habitats. And similarly to my second chapter, I'm just going to dive straight into the results. And if you're really curious about the summary statistics and the model comparison tables, I can show that after this talk. So I will briefly go into each of these, but firstly, PC2 was really only correlated with one variable across all of these metrics, which was strata. And so here, species that are darker live in the understory. And that result isn't new. I talked about that in my second chapter. So we're just going to move past that and instead talk about brilliance, and then we're going to talk about plumage complexity. So for brilliance, what you're seeing here are all the regressions. I've only included slopes if that regression was significant. And what I found is that pretty much identical patterns for both male and female brilliance. I found correlations between climate, land PC2, percent tree cover, latitude, and for females, EVI mean. And what all of this really translates to is just birds in the tropics are darker and birds in more temperate regions are lighter. So birds that live in habitats that are rainier are darker, and birds that live in habitats with more forest cover are darker. And now we're going to talk about plumage complexity. And there's a ton of really rich and interesting detail here that I really don't have time to go into today. So we'll start off with male plumage. And for male plumage, I really only found significant correlations with climate. And I found no direct association between elevated levels of plumage complexity and habitat heterogeneity. And this suggests that sex-specific pressures are likely driving coloration on a per species basis. The significant results I found for males is that males are more colorful if they live in more seasonal climates, and males are less colorful and darker if they live in rainier climates. Females, on the other hand, had multiple significant correlations and really seemed to be influenced by region-specific effects. Both climate PC1 and land PC2 were slightly correlated with female plumage, but had substantially worse AIC scores than the next two competing models, which were mass and standard deviation in visible red. For mass, I found that larger birds are more colorful, but this still did not have nearly as strong of an or as likely AIC score as a model incorporating standard deviation in visible red. And so females with more heterogeneity in visible red are more colorful. And this is a result that really confused me for a while when I first saw it, because I truly did not expect this result. However, the more I dived into my analyses, the more I realized what this metric is truly showing. And the really key distinction here is that this metric is not just describing a spatial pattern, but critically a temporal pattern. Many habitats exhibit cyclical flows of vegetation greenness that varies over the seasons. This flow resembles a sine curve, where half the year a habitat has higher chlorophyll content, i.e. more greenness in its leaves, and the other half of the year it has less chlorophyll content, i.e. more brownness in its leaves. Habitat structures that vary in a predictable manner over the years have these cyclical flows. And those routine changes actually mean there's less variation over the years as habitats return to a predictive norm. And thus, female cardinals have evolved less colorful plumage in habitats that change in a predictable manner over the decades. However, I found that females that live in habitats which vary dramatically across both space and time have enhanced coloration patterns. Species which have the capacity to occupy multiple habitats across their range need to simultaneously balance multiple types of alternative selection pressures. and species that have more homogeneous ranges have much more temporal and stable pressures. And this result shows that elaborate female coloration can evolve in tandem with the compounding pressures of both sexual selection and habitat constraints. And that shows that female coloration is far more complex than previously recognized and really warrants a lot more research. So in summation, I know that was a lot of information. For male plumage complexity, I found that more complex males have evolved in seasonal habitats, and less complex males evolve in habitats with higher precipitation and occupy the understory. For females, I found that females which occupy habitats with higher variation in visible red are larger and are sedentary, have more complex plumage, where females that live in habitats with less variation in visible red, i.e. seasonal habitats, are smaller and are migratory, have less complex plumage. And this result is really important because it shows that species habitats are not monoliths, and habitat heterogeneity exists on a spectrum rather than a binary ultimatum. So because selective regimes can vary dramatically over a single year, we need to develop models which can encapsulate, learn, and predict these fluctuations if we're really going to understand how species respond to these different pressures. And then again, for brilliance, I found that darker birds evolve in habitats with higher precipitation, more forest cover, and if they live in the understory, and lighter birds live in habitats with more seasonality and less forest cover. So in conclusion, I found that habitats are really highly complex and variable mosaics where selection can vary across both space and time. And in order to really understand those processes, we need to be able to capture a lot of that variation. So for males, I found no direct association between plumage complexity and habitat structure. And this, again, means that sex-specific pressures are likely driving coloration on a per-species basis. And this implies that elaborate male coloration is not bound to any specific habitat motif other than the general pattern that darker birds are associated with darker and rainier habitats. For females, I found that coloration is really influenced by these region-specific ecogeographic effects, where highly variable habitats have evolved females to have increased plumage complexity, and seasonal and darker habitats drive plumage complexity to be drab and reduced. And similarly to males, I found that climate and lighter environment drive darker plumage. So in summation across my thesis, I found in my first chapter that UCEs resolve conflicting nodes and establish new phylogenetic relationships within Cardinalidae. For my second chapter, I found that ecology and habitat influence coloration, but the categorical metrics that are commonly used have innate biases within them. And for my third chapter, I found that habitat structure influences female, but not male, plumage complexity. And so with that, I have a ton of people that I would like to thank for this. The first being my thesis committee, and the first one I'll start out with is my advisor, Kevin Burns. I don't think I was the easiest grad student Kevin has had, but Kevin really helped me a lot over the years, and he really pushed me in a way that I needed. And I grew tremendously, not just as a scientist, but as a person with his help. And I truly could not have done this thesis without him. He really devoted just so much of his time to helping me with this. I've never really had someone who has just pushed everything aside for 3 months to help me with something. And I am truly indebted to him for that. I'd also like to thank Allison Schultz, who was really like a second advisor to me and has gone above and beyond what is expected of a thesis committee member. She has helped me so much with statistics and problem solving, and just as someone to talk to, and I, again, could not have done it without her help. I'd also like to thank Todd Reeder for his constant support and help over the years, and just as being a fantastic friend through the course of my 3-year tenure here. And then I'd also like to thank Trent Biggs, who I don't think I would have been able to tackle this monumental 3rd chapter without his help, because he really gave me the theoretical backing to tackle such an auspicious question and really gave me the tools I needed to just take something and run with it. In addition to all these faculty members, I'd really like to thank all of the EB faculty who have each in their own individual way helped me grow tremendously over the years. And many of them don't know the individual impact that they've had, but I can affirmatively say that every single one has empowered me and pushed me to be my best. And I don't think I would be sitting here today to show you everything I've done without their help. I'd like to thank all the museums that I collected this huge amount of data from, my family, which has been incredibly supportive over the years and very necessary to complete this thesis. Then I'd like to thank all of my friends who have really been there, from Rosalind Price Williams, who has really helped me with a lot of the theoretical backing and understanding of phylogenetics, to Dylan Jones, who has really helped me with a lot of the geospatial analyses and just like statistical quandaries. Beyond that, I'd like to thank all of the Burns Lab members, all of which who additionally supported me, as well as my roommates, Ryan and Tony. And then finally, I'd like to thank my partner, Emily Deerdorf, who has been an incredible rock through this entire thesis and has just been incredibly helpful. And I can honestly say that she's probably one of the best things that has happened to me. I'd also like to additionally thank Trent Biggs for introducing me to your graduate student, Emily Deerdorf. Surprise. Thanks, Trent, you're a real one. And with that, I will take any questions that you have.
So take a minute to thank, congratulate you on a really nice presentation. That was wonderful. Finding all the little emojis here. Yeah, great job, Ben. Nice work.
Thank you.
We'll start the question. We'll follow the traditional format. Like I said, we'll start with the committee, then other faculty that are here, and then anybody in attendance. And then once that's done, we're gonna keep the room going, but so the faculty will go into a breakout room and Ben will stay in here. So if any of y'all wanna stay here to support Ben while we're having our little discussion, please stick around for that. Okay, so I guess since I'm already talking, I'll just start with the first question. Would be about, yeah, we've had this conversation before, but I think I wanna talk about it some more. So some of the birds are migratory, some are sedentary. What did you do with the habitats for the birds that, you know, don't stick around in one place? They visit a bunch of different habitats, right? And particularly they have a different breeding and different non-breeding area. And what are the implications of what you did, biases and so forth?
Yeah, that's a very important question and has probably been the bane of my existence for the past 6 months or so. But the plumage specimens that I measured were explicitly for breeding range. And so I needed to only use the breeding range for those specimens, for all my samples really, but especially those samples. And so if I had to basically choose if I was going to use the entire breeding range, the entire range, or subsets. And really, there is no good way to do that without including a bunch of noise in my data set. And so ultimately what I decided is that for those migratory species, I was only going to assess plumage habitat structure in the breeding season. And for the other species, I used the entire year, because if I wanted to model both, I was going to need to incorporate non-breeding plumage, which is data that I didn't have.
Okay, and so would there be some kind, what would be the result of that, or what would there be any particular bias or to that?
I think that there are 8 individuals in the cardinals that have pre-alternative plumage, which means that in different times of the year, they have breeding plumage and non-breeding plumage. And so in order to really tackle that question, I would need to generate an entire new data set. I think that using the breeding range was the best way to mitigate any biases that I could have, because species that are non-migratory have to deal with the same areas and the same selective pressures, while these migratory species are really having those selective pressures in the breeding season. And additionally, that is where those selective pressures should theoretically be the strongest. And that's why I used the breeding range.
Okay, I have like a bunch of other questions, but I feel like I should step aside and let the other committees and other people ask questions at this point. So Todd, Allison, or Trent, if any of you all want to jump in at this point and ask some questions, go for it.
I'll go ahead and, so great job, Ben, and everything.
Thank you.
Glad to, see this. I knew it was going to be good. I'm always sort of interested when people, so you use those concordance factors to kind of look at, because obviously you're taking a multi-gene approach for, for trees and stuff like that, and so. I mean, so what does it mean to you then that, when you talk about having these, like the low, gene concordance factor? I mean, so what is that telling you sort of biologically or evolutionarily here?
Really, it means that the picture is really complex. So having low site and gene and site concordance factor doesn't really mean that that particular split or that node is weak. It's really just an additional metric that can be used to understand the underlying variance across those individual splits. So using, really what it does is showcase how phylogenetic estimates can be biased based off the particular method that you're using, and gives further support for the particular splits that you have.
Okay. Yeah. So I thought you sort of said, so did you say then that just because it has a low value doesn't necessarily mean that it's a weakly supported relationship, or is that?
So yes, gene concordance factor is this decisive number of gene trees that support that particular split, and site concordance factor is the decisive number of gene trees that support that particular split. Okay. And so it does showcase that there's a lot of shared genes if it has very low values. And that means that you need to be really careful when you're using these analyses. But it also shows that because bootstraps can sometimes bias or not paint the full picture, rather, using those metrics gives additional support that you're not just throwing a bunch of data at something and it's giving you the result you want.
So yeah, so I think there's like, it's like just, one section of your thesis you're talking about, because I was trying to get some clarification of kind of what was, what you're thinking and how you're interpreting this, because there was a couple situations where you referred to them having these low CF values as low nodal support, and yet, as you sort of say, those nodes actually had like 100% bootstrap values or 1.0 posterior probabilities. And so are you saying that those maybe nodes are being overestimated by the other methods, or is that just maybe you don't really think of them as nodal support, but just sort of telling you a little bit about the history of the gene trees related to that part of the phylogeny? So I'm just kind of wondering, do you see those as real nodal support, or just it tells you something else about the history of the genes?
I think the latter, that it's really just describing the history of the genes.
Okay.
And I feel confident in all of those splits because I found the same splits across my coalescent-based metrics. And I also found, I didn't present on this, but I used Astral and SVD quartet. So I used 2 different gene summary metrics. So I feel very confident that those splits are accurate, and that, as you said, those gene and site concordance factors are really just painting a better picture of the situation.
Yeah. So do you have any ideas or armchair biology thoughts as to what evolutionary process is maybe giving you that low gene concordance in some parts of the tree?
Oh man, that's a really interesting question, and one where there are a lot of different answers to. So there are a lot of different ways that phylogenetic estimates can be biased, which might generate those results. But in terms of physical and natural processes, I would imagine very recent splits where groups have very shared genetic ancestry would generate a very low gene concordance site factor. And sorry, I'm losing my train of thought for a second. So a lot of processes or reticulation could also be responsible for driving these kind of weird splits that we're seeing.
Okay. Cool. So yeah, so I just have one more question. I really it's about your chapter 3, which I really liked. I really liked what you're trying to attempt there and kind of exploring hopefully a better way as versus using these just sort of broad categorical, characters. I kind of wonder, what do you think about, so obviously there's been a lot of, it's really cool that you kind of showed that over a 10-year cycle or even a seasonal cycle that obviously the habitats, can change due to succession, seasonality or whatever. But what about over multiple decades, like habitat deconstruction, destruction and stuff like that? And so that you maybe have some of these birds living in habitats that maybe really isn't their ideal habitats anymore, or maybe aren't the habitats that actually maybe influenced the plumages they have now. I'm just kind of wondering that lag sort of between those species that maybe are living in habitats that aren't really the ones that maybe influence the phenotypic difference you see there. Do you think that's a problem or not, or a real thing, or what's the?
I think that that's a very valid concern. I would say that at the end of the day, all of the ways that we categorize contemporary habitat are subject to those results, because climates vary dramatically over the millennia. And so really to tackle that question to its full need, I would need to generate ancestral niches and then incorporate that. In terms of how habitat degradation can influence these results, I would say that it is possible that those values can be influenced, but I would also, I think that not analyzing these metrics, using these more quantitative analyses is worthwhile. So we might have this minor effect of habitat degradation across their entire range, but I would argue that that effect is mitigated by the biases that we're introduced when we're using categorical metrics.
Thank you. And I agree with you. I think it's better to do these things versus worrying about all the little concerns and all that. So good job. So I don't have any more. I don't have any questions.
Thank you.
I'll ask a question next. First of all, great job, Ben. Really fun to see this all come together. So my question for you is, you've taken 2 incredibly complex traits for each species, right? You've got these, plumage color, which is a sum of color over many different patches, over many different types of measurements. Then you've got climate, which of course varies, as you said, both temporally and seasonally, and come up with a few descriptors of each of these complex traits. So I was wondering, if you're thinking about plumage specifically, how do you think your results might have changed if you considered plumage not as a single whole for each species, but for example, maybe looked patch by patch or looked at some of the components that make up plumage color?
That is a question I was really trying to jam into this thesis, and I ultimately just needed to step away. Basically, plumage is a composite trait, so it's comprised of a lot of different variables. And anytime you use dimensionality reduction, you're going to be removing some of that signal. When I regressed the individual traits, I found very similar patterns, but I've not tackled those individual traits, and I think that that would be a really interesting future study, because if I can find correlations between, say, patches that have more carotenoids versus patches that have higher UV reflectance, that would really paint a really interesting framework for how these patches evolve, and give, I think, a much more robust understanding for how these processes originate in the first place.
Yeah, thanks, but I agree. Lots of future work to be done with these data sets that you've gathered for sure. And we've already talked quite a bit, so I'll leave it for others to ask questions now.
Okay, hi Ben. Phenomenal job. I mean, it's amazing to see how you pulled together these different parts and made a coherent story out of it. Congratulations, and you know, very impressive.
Thank you.
Yeah, and I will say he took, you know, he would Ben would come into my office for a half an hour and ask some great questions, and I'd toss out some ideas, and then he'd come back a month later and said, I did everything, every suggestion that you had. I was like, oh my God, that's amazing, right? So, so phenomenal set of technical skills that you developed along the way. So I think chapter 3, like everybody's saying, is super interesting, and I wonder if I think I tended to lose some of the crispness of the story when the climate is and land use is summarized as principal components, and you might be able to go back and generate almost as few metrics that describe the climate, because you ended up finding out that all those 17 variables collapse down into rainfall, basically. So like mean rainfall and seasonality, maybe temperature, and that might make the story stick a little bit easier in the readers, for the reader. That's just one suggestion for, you know, sort of instead of using PC for climate and even for land cover, go back to more sort of directly intuitive summary metrics. And we can talk about that, but Emily can tell you about the aridity index, for example. You know, that might be helpful there. And then, oh, so if you mentioned the limited Google Earth Engine processing time, if that was not a constraint, would you be able to better answer Kevin's question about the impact of spatial variability of habitat occupancy and space and time, so that you could so that you could weight your NDVI value by probability of occurrence and duration of occurrence, something like that, that might also crispen those results up.
Absolutely. There are a million other things I wanted to do on this data set. So if I did not have that computational limits, I could actually use the species distribution models and create TIFs. And so then I could actually weight them by where the true most probable areas they are, and then span outwards. One thing I was also playing with is using like Fourier transforms. So instead of having Google Earth Engine generate these summary statistics, I captured all that data and used a Fourier transform to understand the actual like spectral frequency of how it's changing. I couldn't really deduce that down into a comparative framework. And then the other metric I would be really excited about is something called zeta diversity, which is really similar to other metrics like alpha and beta diversity, except it's accounting for the size of their range and weighting it by that. So that long-winded answer was basically, I have a million different ways that I want to analyze this, and I ultimately just had to settle for the way that I was doing for the purposes of this thesis.
Yeah, that's great. Totally makes sense. And that said, with the my comments aside, I mean, I think that chapter's very close to publishable as it is. I don't think you need to toy around with it too much more. There's a few things that you might try again with these more direct climate indices. But other than that, it's very, I think it tells a great story. And from my perspective as a hydrologist, I don't know what the actual bird or ornithology community, I'm sure they're also interested because it's so unique. But yeah, so thanks. I'll leave it there.
Thank you.
Okay, if there are no more questions from the committee, are there questions from the other faculty that are present? See, Andy's here, Jeet's here. Any other faculty have questions? I saw a hand, I think. Oh, Jeet's got his hand up. Jeet, go for it.
Hi. Oh, looks like my camera's burned out again. COVID's taking its toll. Sorry. But great job, Ben. And as with everyone, they're all great chapters, but chapter 3 is particularly exciting. We pulled together a lot of different threads of evidence and tied them together in a cohesive framework. And I'm excited not just about the results, but that chapter standing as a foundation for an entire research program. But lots of little questions. I've got some interesting side project analyses which we can talk later on about. But one thing that I thought would be pretty cool is if you could automate your classification system, your quantified habitat classification system. That'd be great, because then you can export that to so many other groups can use it well beyond, you can plug in a species, get the data and get that. And what I think would be really cool is if you go back to your original sources of human curated habitat categories and see how those compare to your quantified things, your quantified habitat categories. You think that's possible?
Yeah, I think that's a really great idea, actually. I think that when I go to publish this, I really want to paint the picture of how these categorical metrics bias our results. So I was really hoping to kind of include that data and make it into a single analysis, or a single paper rather. But yeah, I think that that's a phenomenal idea to include.
Okay, no, I have lots of little questions, but we can talk about later. The other big project that maybe if you want, when everything settles down and maybe after you get tenure and you want to revisit this question, we can talk about working in historical processes and asking if some of these traits, how they influence other aspects of the evolution process, whether it's migration, a causal relationship as opposed to a correlative relationship, a process-based approach.
I would love to do that. I'm very interested in exploring this beyond correlative metrics and diving really deeper into the statistics of it.
All right, so that's all from me. Thanks. Good job again.
Thank you so much, Jeet.
Looks like, thanks, Jeet. Looks like Andy has his hand up. Andy, you have some questions?
Ben, I'm going to pay you a great compliment. I'm interested in learning more about some birds.
Nice.
Which I don't know that I've ever said, at least not out loud. Is there an easy way to pull up a slide without disrupting everything?
Yeah. Is there a slide in particular?
I like the second part of the talk, maybe the second to last correlation slide.
With all the correlations?
No, it was just two. So it was like second to last slide in that part of the talk.
Sorry.
Two
For
punch
my
lines.
Second or third chapter?
Second.
Oh, I know the slide you're talking about.
Was there a data point misplaced in the figure?
I don't know. Nah, let's see, go.
Come on.
Oh, okay, sorry. Sluggish.
Yeah, this is on my old computer and it is not super happy. It's fine, whatever. Not this one?
No, the scatter plot with the correlation.
Oh, this one.
Yeah. So what like in the PC1 plot? What's the leftmost point? What is that?
That is Cynolaxia cyanocodes.
You have to give me something more. What tell me about that.
Oh, the leftmost plot is a species that is exceptionally dark compared to the rest of them. It's
Where
very
is it
dark.
found?
It's found in the tropics from just above Costa Rica down into around Colombia.
Okay, and what about the PC2 plot? What is the what's the leftmost point on the PC2 plot?
That one is, oh my God, I butcher this genus so often. Cyanothorax cyanocades. And it's found in kind of more northern regions, kind of near the Yucatan, but still central Mesoamerica.
Okay, so when I look at these plots, those 2 points on each one have extremely high leverage, right? Like there's a good chance you pull out 1 point on the left plot, 1 point on the right plot, and nothing significant anymore. To me, that's the story, is like, what is it about those species? What is it about their distribution, their biology, their habitat? Like they seem to be driving a lot here on these 2 overall patterns. And so my suggestion for future work, for writing up a summary and a paper is that's what I'm most interested in, is those 2 points, those species, what they are and what they represent.
I'm very glad you brought that up, actually. I have actually removed those points because they seemed like outliers to me. They didn't actually test as outliers. I ran an analysis that was basically seeing if any individuals were the residuals of the PGLS were plus or minus 3 standard deviations away. And none of them tested as an outlier, but you're really diving into what constitutes an outlier at this point, and that is a whole body of literature. I did analyze them with and without those external points, and my results didn't really seem to change.
Right. Again, I've got less interest in having a sort of discussion about the outlier, and more about like the biology and the distribution of those species specifically, and what they represent in terms of the overall pattern.
In terms of the overall pattern, I think it basically just paints a picture for PC1 that darker species are darker together with less complex plumage. And as we move out, species do tend to kind of bifurcate, but we still see this consistent pattern where highly colorful males are generally related with highly colorful females. And that is because of their shared genetic architecture. And that's also a result that, regardless of the habitat metrics, has been routinely proven across most metrics that analyze the evolution of color of male and female species. Does that answer your question?
Yes, thank you.
Any other faculty present that have any questions? Speak now, raise your hand. Did you have another question, Jeet, or that's left over?
No, is my hand still up? Oh, no, I'm good.
Okay, anybody else? Let's find out something that we haven't thought of. Ben, what do you got? Amelia's raising her physical hand.
Yeah, Ben, that was a phenomenal talk. It's really awesome to see the results of so much hard work. So Rosalind Price-Waldman and I were bouncing around this question. It's about the question you posed to the audience about how human bias influence what goes into environmental reconstructions for your work. So I wrote it down. So how has it influenced kind of past environmental reconstructions? It's like bias you were talking about. Does your pipeline have any caveats that still create bias, and what matters for reconstructing a cardinal's environment across space and time? And what steps can future projects take to be more comprehensive?
Oh, which one do you want me to answer first?
I'll paste it in the chat so that you can read it, because I know, but it was just so profound. It was really, really nice.
I would say that I reconstructed the ancestral states. I love that it starts with a great talk and that you wrote that out. I reconstructed the ancestral traits of all of these categorical metrics. And really what I was saying was just that forest of some kind was their ancestral state. And as they bifurcated out, it seemed like more colorful species evolved in habitats that were more semi-open. In terms of these biases, I think that whenever you are trying to reduce something that is as incredibly complex as a habitat, you have to make a lot of assumptions. And using these categorical metrics are a really great way to paint a picture of these general patterns. But a lot of times they can really fail to delineate the true habitat structure. And I think a great example of this is in Southern California, we have oak woodlands, which are not quite forest and not quite grassland. And so depending on who is coding those traits, they might choose one over the other. And in doing so, it creates this, this bias of how you kind of visualize what a habitat is. And through that process, you are assuming something about how the bird has evolved. So I think that a lot of phylogenetics is like summarized in doing our best to reduce these really complex pathways and processes into an understandable and cohesive framework. But it seems that sometimes when we use those, how we kind of define it creeps in and might artificially inflate our results.
So the way that you've defined it, was that a step forward? Is that just enough of a step forward for future studies? Like, where do you see this going?
I see this going that really when we're analyzing habitat, we need to not just analyze it in terms of where they live, but how the habitat changes over the years. Because you can say something is seasonal, but how it is seasonal is a really important metric. So I don't think that those categorical metrics are useless. I think they're really important for painting a very broad picture of a process. But going forward, I think that if we really want to answer this question, we are going to start to need to include all these different aspects of habitat.
That's really cool. Congratulations. Thanks.
Thank you.
Thanks, Amelia. Anybody else have questions for Ben? Oh, Phil Unit. Good to see you, Phil.
Thank you so much for inviting me. Very interesting. Thank you.
Great to see you, Phil.
One aspect of plumage variation in the Cardinalidae is the rate at which the males acquire their definitive plumage. Some yearling males are female-like, some are variably intermediate, and some achieve definitive plumage in their first year. So do you see any association between this aspect of variation and habitat variation?
That's a really interesting and I think a really important question that kind of comes back to this concept that I think in comparative methods, or specifically color evolution with birds, it's really only recently that we started to understand the importance of molt and how it influences these different patterns. And the data set that I talked about, Guiar et al., that used this habitat metric, was quantifying how molt influences, how different habitats drive different molt patterns. I am a little dubious of some of the statistics and the compared methods of that paper, but I think that it still showcases that different molting strategies are correlated to habitat structure. In terms of my results, I'm not super worried about the immature males because they're not likely breeding. So the effects that I am testing are probably not applicable to individuals. And then I hopefully mitigated the bias by only using the breeding range. But again, until I can get a data set that I can encapsulate that non-breeding range, I won't fully be able to answer it. But I will say that I have a collaborator at Occidental College, and we're working on trying to build that data set and trying to analyze this in a more robust framework.
Thank you.
Thank you for your question.
Any other questions? Just raise your hand or just speak up. Going once, twice. Okay, so let's give Ben another round of applause.